Dear students, welcome to KPS Bio Tutorials. Today I would like to talk about a topic, namely glycolysis. Glycolysis is a part of metabolism, metabolic reactions. We know that the metabolic reactions are the whole set of reactions taking place in living cells, and which is necessary for the maintenance of life. So it is one of the unique feature of living things. So these metabolic reactions are broadly classified into two categories: the anabolic reactions and the catabolic reactions. Anabolic reactions are the synthesis of larger compounds from smaller compounds. For example, synthesis of protein from amino acids, synthesis of starch out of glucose. These are examples for anabolic reactions. That means synthesis of larger compounds from smaller compounds. Whereas the catabolic reactions are the breakdown of larger compounds into smaller compounds, breakdown of proteins into amino acids. So these are the two categories of metabolic reactions: anabolism and catabolism. Among these, the glycolysis is a part of carbohydrate metabolism, and as the name indicates, glycolysis means glycolysis, that means breakdown of glucose. So today we can discuss in detail about glycolysis. Once again, welcome back to today's topic, that is glycolysis. It is also called Embden-Meyerhoff's pathway, or EMP or Embden-Meyerhoff pathway. As the name indicates, glycolysis is the breakdown of glucose. Here, a 6-carbon compound glucose is broken down into two 3-carbon compounds, namely pyruvic acid or lactic acid. Pyruvic acid may be later converted into lactic acid. So actually glycolysis is the breakdown of glucose into two molecules of pyruvic acid. Here glucose is cut into two pieces. Exactly, the cutting occurs at the middle of the compound. That is why the 6-carbon compound glucose is broken down into two 3-carbon compounds, namely pyruvic acid. You have been studying glycolysis from your +2 standard onwards, or even from secondary standards. It is occurring in 10 steps, and the site of glycolysis is the cytoplasm. It is taking place within the cytoplasm. There are 10 steps, that means there are 10 intermediate compounds. Among these 10 intermediate compounds, 9 intermediate compounds are phosphorylated compounds. The phosphorylated compound means a compound with a phosphate group or phosphoric acid group. This phosphoric acid group is a negatively charged compound. So when the phosphate is added to a compound, the compound itself becomes a charged compound. The importance of this charging of compound is, once the compounds are charged, the cell membrane becomes impermeable to that charged molecules. So once a compound is charged within the cytoplasm, then the compound cannot escape out of the cytoplasm or out of the cell. So in order to trap all the intermediates of glycolysis within the cytoplasm, these compounds are get charged. Then the entire process of glycolysis is divided into two equal half pathways. One is called phase 1 and other one phase 2. Glycolysis is taking place in two phases. One is phase 1 and other one phase 2. The first 5 steps of glycolysis are coming under phase 1 and the remaining 5 steps are coming under phase 2. Phase 1 is an energy consuming phase. That means here 2 ATP molecules are consumed during this phase 1, and in phase 2 it is called payoff phase because 4 ATP molecules are synthesized in this phase. Today I would like to talk about the phase 1. It includes the first 5 steps of glycolysis. So let us go to the steps of glycolysis. The first step, all of you are knowing about these steps. First step is the phosphorylation of glucose, or here the glucose is activated into glucose 6-phosphate using one molecule of ATP. Here one ATP is hydrolyzed and the one phosphate is attached to the 6th carbon atom of the glucose. Here you can see the 6th carbon atom of the glucose. Into the 6th carbon atom of the glucose, the phosphate group, phosphoric acid group is attached and the ADP is freed. Here the enzyme catalyzing this reaction is hexokinase with the help of the cofactor Mg2 plus metal ions. Hexokinase is an important enzyme, as the name indicates. It is a kinase enzyme. That means it utilizes ATP for phosphorylation and it phosphorylates all hexo sugars. Thereby it is called hexokinase. The hexokinase enzyme is of two types, type 1 and type 2. Type 1 is present in brain and type 2 present in skeletal muscles. These are different isoforms of the hexokinase. They are present in different tissues. Liver contains both type 1 and type 2 hexokinases. The hexokinase enzyme is inhibited by the excess glucose 6-phosphate. Here the product itself is inhibiting the enzyme by feedback inhibition. So it is acting as a regulatory mechanism for hexokinase. If excess glucose 6-phosphate is synthesized, automatically the hexokinase is switched off. There is a similar enzyme, namely glucokinase, present in the liver, which is present in abundance in liver. It is also catalyzing the same reaction, that is phosphorylation of glucose into glucose 6-phosphate. But the glucokinase present in the liver has lesser affinity to glucose. It has high Km value when compared with the hexokinase. That means it has a lesser affinity to glucose. That means it will phosphorylate glucose into glucose 6-phosphate with less efficiency or with less velocity, reaction rate. The rate of the reaction of phosphorylating glucose into glucose 6-phosphate will be lesser when compared with that of hexokinase. And also the glucokinase is not inhibited by excess glucose 6-phosphate. Even in the presence of excess glucose 6-phosphate, the glucokinase will continue its reaction. That means the usage of this glucokinase is in the synthesis of glycogen. The first step in the glycogen synthesis is also phosphorylation of glucose into glucose 6-phosphate. So excess glucose in the liver or excess glucose in the blood is converted into glycogen in the liver with the help of this glucokinase enzyme. So that is why it is not inhibited by excess glucose in the glucose 6-phosphate. So from these we can understand that the glucokinase is acting at the excess when the glucose kinase will be activated, or its action is taking place when there is excess glucose in the blood, whereas hexokinase is a housekeeping enzyme. It is always will be active. So when there is a need of ATP synthesis, the hexokinase will be activated. The glucose 6-phosphate, the product of the first step of glycolysis, that is glucose 6-phosphate, it is said to be a metabolic junction because the glucose 6-phosphate has so many fates. It may enter into glycolysis and complete the glycolysis, or it may enter glycogenesis or the synthesis of glycogen, or it may enter the HMP pathway, or it may enter gluconeogenesis. So there are so many fates for glucose 6-phosphate, thereby it is called metabolic junction. Then second step of glycolysis is the glucose 6-phosphate is isomerized into fructose 6-phosphate. Here glucose is an aldose sugar, fructose is a ketose sugar. Here aldose and ketose interconversion is taking place. The enzyme catalyzing this reaction is Phosphogluco Isomerase. Here you can see the structure of glucose. Mostly the glucose is existing in the pyranose form, whereas here it is in the furanose form. It is a ketose sugar. Fructose is a ketose sugar. Here glucose 6-phosphate is isomerized into fructose 6-phosphate. So both are having a phosphate at the 6th carbon atom, and the enzyme catalyzing this reaction is Phosphogluco Isomerase. The cofactor for this enzyme is Mg2+. So this, as the arrows indicate, it is a reversible reaction. It is just an isomerization reaction. Then the third step is fructose 6-phosphate is again phosphorylated into fructose 1,6-biphosphate. Here the fructose already have a phosphate at the 6th carbon atom. It is again phosphorylated at the 1st carbon atom also to form fructose 1,6-biphosphate. Here one ATP is utilized. Using the energy of ATP hydrolysis, phosphate is added to the 1st carbon atom of the fructose 1,6-phosphate with the help of the enzyme phosphofructokinase, or simply PFK. This is the most important enzyme in glycolysis, that is PFK or phosphofructokinase. This step of glycolysis is said to be the committed step of glycolysis because here as the arrow indicates, it is a irreversible reaction, and also the fructose 1,6-biphosphate has no any other fate other than glycolysis. Once the fructose 1,6-biphosphate is formed, it has no way but to enter the glycolysis. That is why it is called committed step of glycolysis. The enzyme catalyzing the committed step of glycolysis is phosphofructokinase. Thereby this enzyme is called the pacemaker of glycolysis. PFK is said to be pacemaker of glycolysis. So PFK will be active when there is a need of synthesis of energy or ATP, and if there is excess ATP, the excess ATP allosterically inhibit PFK, because excess ATP means there is no need of further continuation of glycolysis. Thereby, the ATP itself acts as an allosteric inhibitor for PFK, and by its binding, it will lower the affinity for the fructose 6-phosphate. PFK's affinity for fructose 6-phosphate will be lowered by excess ATP. Whereas the AMP reverse the inhibitory action of the ATP. If there is excess AMP in the cell, then it will reverse the inhibitory action of ATP, and thereby the PFK become active and glycolysis continues. Here in this graph, you can see the activity of PFK at different conditions. As the concentration of fructose 6-phosphate, it is the substrate for the PFK. As the concentration increase, usually the enzyme activity also increase up to a particular level. But at low ATP level and high pH, even at the low concentration of the substrate or fructose 6-phosphate, the PFK activity will be very high. You can clearly see in the graph. Whereas at high ATP level and low pH, the reaction rate is much slow. If there is high ATP, that means there is no much need of energy synthesis. That means no much need of glycolysis. Thereby this enzyme becomes slowed down. And also at the low pH is produced because of the accumulation of lactic acid. So if there is excess accumulation of lactic acid, that indicates there is a lot of energy is already produced, a lot of glycolysis activity is already done. So thereby the pyruvic acid is converted into lactic acid. So that also indicates the presence of high concentration of lactic acid, also indicates there is high level of energy in cells or blood. Thereby the PFK activity is slowed down. Then the fourth step of glycolysis is the fructose 1,6-biphosphate is broken down into two 3-carbon compounds, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Fructose 1,6-biphosphate is a 6-carbon compound which is broken down into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Both are 3-carbon compound, whereas glyceraldehyde 3-phosphate, glyceraldehyde is an aldol sugar, dihydroxyacetone is a keto sugar. So here one 6-carbon keto sugar is exactly divided into two equal parts. One from the glyceraldehyde 3-phosphate, other from the this is the dihydroxyacetone phosphate. This one is glyceraldehyde 3-phosphate. Here, in the fructose 1,6-biphosphate, there are two phosphate, one at first carbon and other one at the 6th carbon. When it is cut into two pieces, here one phosphate goes to glyceraldehyde 3-phosphate and the other phosphate goes to dihydroxyacetone. Then the fifth step is the dihydroxyacetone phosphate is again isomerized into glyceraldehyde 3-phosphate by an enzyme namely triose phosphate isomerase. So it is also a simple aldo-keto isomerization reaction. Here the dihydroxyacetone phosphate is a keto sugar which is isomerized into glyceraldehyde 3-phosphate, which is an aldol sugar, by an isomerase enzyme triose phosphate isomerase. And thereby at the end of the fifth step of glycolysis, the glucose is broken down into two molecules of 3-carbon compound, namely glyceraldehyde 3-phosphate, and these 5 steps are altogether called the energy consuming phase. In this phase, 2 ATP molecules are utilized, one at the first step for the phosphorylation of glucose into glucose 6-phosphate and the other one in the third step, that is for the phosphorylation of fructose 6-phosphate into fructose 1,6 biphosphate. So totally 2 ATP is hydrolyzed into ADP, and the energy out of this hydrolysis is utilized for the phosphorylation of the glucose and fructose. So this is all about the first phase 1 of glycolysis. I hope all of you understood the topic very well. Thank you for listening.